Hi everyone, today in regards to our article, we're going to be introducing a really new and exciting scientific discovery about cell membranes and giving some examples to demonstrate their amazing capabilities of cell membranes.
In July of 2016, scientist Chen and his researchers have created a new thin material that mimics cell membranes. Such a material has long been sought for applications in water purification and drug delivery.
Researchers in this article mention that cell membranes are selective about what they let pass through, using tiny embedded proteins such as transport proteins as gatekeepers. This means that the cell membranes are selectively permeable. Membranes repair dings to their structure automatically and change thickness to pass signals from the outside environment to the cell's interior, where most of the action of the cell occurs.
By using 4 bendy straws and a bubble-like solution, we were able to demonstrate the characteristics that scientist Chen and his team mimicked when recreating their material to improve water purification and drug delivery. The scientists in the experiment were able to recreate cell membrane permeability and its gatekeeper-like qualities. As you can see, when I bring the pencil to the bubble, it pops. That is because objects that are too large, charged, or not the same material as the cell membrane cannot pass through on their own. If I were to soak the pencil inside of the bubble solution, the pencil will be able to pass through without damaging the membrane. This is because objects that are the same material, small and uncharged, can pass through easier without damaging the membrane. If we take a small plastic tube, it can act as a transport protein on our bubble membrane. Since large and charged molecules cannot pass through on their own, the transport protein helps assist them while diffusing through the membrane, causing no damage to the cell membrane itself.
Scientists would like to take advantage of membrane properties such as gatekeeping to make filters or signaling to make sensors. A cell membrane-like material would have advantages over other thin materials such as graphene. For example, mimicking a cell membrane's efficient gatekeeping could result in water-purifying membranes that don't require a lot of pressure or energy to push the water through. Researchers describe lipid molecules as long and mostly straight. They have a fatty end that prefer to hang out with other fats, and a water-like end that prefer the comfort of water. Because of this chemistry, lipid molecules arrange themselves with the fatty ends pointed toward each other, sandwiched between the water-loving ends pointed out. Scientists call this a lipid bilayer, essentially a sheet that envelopes the contents of a cell. Proteins or carbohydrate molecules embed themselves in the membranous sheet.
There are 2 categories of proteins within the cellular membrane. The first are transmembrane proteins and integral proteins, which are embedded into the cellular membrane. Some integral proteins span the entire membrane, while others do not. Peripheral proteins are loosely bound to the exterior of the cell membrane.
Glycolipids are lipids with an attached carbohydrate. They are found on the outer surface of eukaryotic membranes. They provide energy and provide a standpoint for cell recognition.
Cholesterol is a type of lipid that aids in maintaining the fluidity and overall integrity of the cell membrane. It also regulates temperatures and the secretion of certain proteins. The selectively permeable nature is carried out by the cell membranes. It's defined as a fluid mosaic model. It's a mosaic in the sense that it's composed of phospholipids, proteins, and cholesterol and glycolipids. It's fluid in the sense that it's always moving in a constant bilateral motion. The phospholipids, which are the fats, are amphipathic in nature, in that the dual nature between the phosphate-rich hydrophilic heads and the 2 fatty acid tails, which are hydrophobic. They allow for this transport of small and uncharged particles. Proteins allow for the transport of large and charged particles, and they're embedded within the layer.
After creating a lipid-like peptoid and trying several different methods, the team found that after putting the lipid-like peptoid into a liquid solution, the molecule spontaneously crystallized and formed what the scientists call nanomembranes, which are straight-edged sheets as thin as cell membranes.
The team simulated how single peptoid molecules interacted with each other using a molecular dynamics software. To test whether their synthetic membranes had the signaling ability of cell membranes, the researchers added sodium chloride salts. Salt makes real membranes thicken up, which explains why as the researchers added more salt, the thicker the nanomembranes became.
The team was also successful in making their membranes self-repair and undergo osmosis.
The results concluded that Chen and his team were on the right track towards fostering new materials that mimic cell membranes.